Thanks. So good afternoon. Indeed, my name is Edith, and I'm hugely honored to join this prestigious academy. And I really look forward to working with all of you in the future. And I'm very sorry not to be there with you today. I'm currently the Director General of the European Molecular Biology Organization and a molecular geneticist by training. I actually started my scientific life as a physicist. In fact, I was an aspiring astronomer, but I turned to biology only when I went to university in Cambridge, where rather than looking up at the stars, I decided to look into the beautiful, complex life on our planet. I became fascinated by the life sciences and the incredible wealth of opportunities that the technologies of the last decades had provided in order to understand the molecular basis for life. It was actually so exciting, inspiring, and felt so useful to work on life itself that I decided to dedicate myself indeed to the understanding of biology. So after a PhD in cancer, in fact, in cancer genetics in London, I decided to move to the Pasteur Institute in Paris as a postdoc in the laboratory of Philippe Avner, where I started to work on X chromosome inactivation, which is a remarkable process that concerns how one of the two X chromosomes in female mammals becomes silenced during development. So females have two X chromosomes, males only have one. And the way to achieve balance of gene expression between males and females is to shut down one of the two X's in females early on. In fact, females are actually cellular mosaics for their X chromosome expression due to this process. And Mary Lyon, who was a remarkable woman, a geneticist who discovered this process back in 1961, in fact, actually described this whole process thanks to mouse genetics and looking at phenotypic diversity that she could see. I was very lucky actually to have met this most inspiring scientist. So this process is actually essential for life. The X chromosome is very large, and the improper expression of genes on it can have dramatic consequences. So it makes it relevant to diseases such as cancer, where epigenetic processes are disturbed, as well as autoimmune syndromes that are more prevalent among women. It's also a fascinating process from the molecular point of view, as it's a paradigm for epigenetics. The two X's are treated differently in the same cell, in the same nucleus, and the differential treatment is actually propagated stably through cell divisions. So the big question is, how is the genetic material marked to be silent on one X chromosome through epigenetics, and how is this silencing also reversed? And so both as a postdoc, and then when I set up my own lab in 2001 at the Institut Curie in Paris, I've been exploring with my group how X inactivation happens, its molecular mechanisms and its dynamics in development, physiology, and disease. So when I started my work on X inactivation, very little was known. And I was in one of the labs, I was lucky enough to be one of the labs that helped to uncover the very first molecule that triggers this process during development, a long non-coding RNA that covers the chromosome and triggers its silencing. So this led me down many paths, exploring the study of RNAs, of epigenetics, of stem cells, and embryology. And one of my lab's first major advances was to show that epigenetic changes can be incredibly dynamic, particularly as an embryo forms and matures. We described the dynamics of this process by looking in both mouse, rabbit, and human embryos, as well as stem cells. Our work also provided some very fundamental insights into how the genome is organized in the nucleus, and we co-discovered that this organization corresponds to what are known as topologically associated domains, or TADs, which correspond, in fact, to the regulatory landscapes of genes in many cases. And most recently, our research has revealed the epigenetic plasticity of X inactivation in somatic cells, where some genes become reactivated or escape X inactivation and expressed in a double dose. And this means that this activity can have a tremendous impact on individual female physiology and variations in this, as well as important implications for sex bias in disease. So most of my career has in fact focused on epigenetics, and X inactivation is a big example of this. Now, this is a word, a term that has had many definitions and is often, I would say, a trigger of both hopes and fears. Are we more than our DNA? Is DNA not our destiny? Or, for example, the fact that severe stress or trauma has been proposed to have an impact on future generations. Is epigenetics involved? Are there epigenetic mechanisms? So in some ways, epigenetics is now very loosely used to describe links between genes and the environment. And in fact, the word, the term, the discipline has its origins in epigenesis as well as genetics. And epigenesis was the concept that went as far back as Aristotle, who described how development consists of the emergence of complexity from simplicity, from a fertilized egg to a complex organism. It was actually Conrad Waddington, an embryologist, philosopher, paleontologist in the 1940s, who decided to coin the word epigenetics to describe the link between genes and the control they have of development and the environment, in this case, talking about the embryonic environment, to explain how epigenesis happens. So my own work on exon activation actually falls into the more specific definition of epigenetics as the study of molecules and mechanisms that can perpetuate alternative gene activity states in the context of the same DNA sequence. This is as cells divide or even across generations in some organisms, such as plants. It was, in fact, when I became a professor at the Collège de France in 2012, and I started to lecture to the general public on the topic of epigenetics. So every year I had to give annual lectures on different aspects of epigenetics, that I began to explore this topic in a wider context, and particularly to think about the environment and the impact that the environment can have on the variation in gene activity and in phenotypes in our cells and tissues. And also how this can contribute to biodiversity, even within a species. The role that epigenetics has in buffering against environmental stress or in enabling plasticity in gene activity as a result of environmental changes. It was this, I would say, exposure to new horizons that led me to take on the job that I currently have, which is Director General of the EMBL. This is an organization, it's the only organization for life sciences research in Europe that is intergovernmental, and it's a collaboration among 28 European countries. And since I joined in 2019, we have been able to set up a new program that explores life in its natural environment. Particularly at this time of planetary challenges, EMBL's new program seeks to rise up to some of the challenges and find some solutions using molecular tools and molecular biology. And it's a perfect example of collaboration, of open science, and of training the young scientists of the future. So EMBL is actually situated in 6 different institutes across Europe, one of them being in the UK, another in Heidelberg, which is where I am located, the headquarters. And I made this choice to lead Europe's unique intergovernmental organization for life science research for many reasons. The main ones being that I deeply believe in fundamental curiosity-driven research. And I also believe that science should be done without frontiers. I'm actually of half Greek, half English origin. I was raised in the UK and I now live in Germany and in France. I'm a true European, but also a deep believer in intellectual freedom. And in this context, and perhaps maybe relevant for my future interactions also with the Academy, I was one of the founders of a program in France called Pause, which is to help scientists in exile who seek scientific freedom in France, fleeing from countries in conflict zones. So just to say one last word about EMBL's new program, which we've called Molecules to Ecosystems. This really seeks to explore new frontiers to understand life in its natural context and how organisms respond and adapt to changes that are either natural or man-made, including climate change. In other words, how life evolves, but also how it survives. So this is a challenge that we have to confront more and more. And we couldn't really measure environmental changes in the past, at least at the molecular level, but today we can. We really do have the tools in biology. And if we really want to understand life, we have to actually go out there and understand how these kinds of environmental fluctuations have an influence on biodiversity. What is the impact of pollution? What is the impact of climate change? And how do communities of organisms behave together when they're confronted with new pathogens such as the virus that we've been confronted with in this pandemic? So in this context, I hope that my role, not only as a scientist in epigenetics, but also as the Director General of EMBL, I'll be able to participate actively in the Academy. And I thank you so much for having elected me.